Good morning, I'm Julie McNicholas.
And I'm Erin Cromwell.
And we are doing our presentation with Samantha Ward on Sir Martin Evans, the winner of the 2007 Medicine Physiology Nobel Prize.
Sir Martin Evans was born in Stroud, UK on January 1st, 1941. Alright, throughout Sir Martin Evans' childhood, he got sick a lot, so he wasn't present in school, but he still did a lot of learning on his own and kept up with schoolwork even though he wasn't there. So from early on, Sir Martin Evans had developed a love for biology in high school. He dropped math class so that he could take extra classes in chemistry, zoology, and botany.
In 1959, he won a major scholarship to Christ College in Cambridge, where he continued to study zoology, botany, and chemistry, but later dropped zoology and added biochemistry. This was important because it was around the same time that huge advances were being made in the field of genetics. Sir Martin Evans found himself drawn to plant physiology and function during his time at Christ College, and greatly enjoyed attending seminars and lectures by both Sidney Brenner and Jacques Monod. An interesting fact about him is that he did not take his final examinations at Christ College because he was sick yet again with glandular fever.
After college, Sir Martin Evans decided on a career examining genetic control of vertebrate function. He moved to the University College London, where he did research and taught graduate and undergraduate students. While he was at University College London, he learned laboratory skills from Dr. Elizabeth Ducher. So Sir Martin Evans was awarded his PhD in 1969. Later, he became a lecturer of anatomy and embryology, which is interesting because he didn't realize that embryos would become a huge part of his life.
After receiving his PhD, Sir Evans made it his goal to research how to isolate developmentally controlled mRNA and moved from the University College of London to the University of Cambridge in 1978 to work in the Department of Genetics and pursue this idea. In 1980, Sir Evans began working with Matthew Kaufman at the University of Cambridge, and they worked to discover how to use blastocysts for the isolation of embryonic stem cells, or ES cells.
After Matthew Kaufman left the University of Cambridge, Sir Martin Evans started taking many different paths. As he says, he was drawn into a number of fascinating fields of biology and medicine, so he kind of experimented around, saw what he liked. Eventually, he ended up going to the Whitehead Institute in Cambridge, Massachusetts for a month, where he practiced and learned the latest laboratory techniques to take back to England. In November of 1985, Sir Martin Evans returned to the University of Cambridge in the United Kingdom to continue his work on stem cells.
In 1999, Sir Martin Evans began working as professor of mammalian genetics and the director of the School of Biosciences at Cardiff University. The School of Biosciences is now named after him due to his award for the Nobel Prize.
Sir Martin Evans' first experiment trying to isolate developmentally controlled mRNA didn't work out for him, so he moved on to his next experiment, which was trying to modify cancerous cells of the ovaries and testicles so that he would be able to modify the DNA of the offspring, but this didn't work out for him because cancer is really unpredictable and dangerous, and there wasn't enough technology at the time, so he wasn't able to modify the offspring.
At the time of his second failure, Sir Martin Evans began working with Matthew Kaufman yet again, and this was also the same time that it was discovered that it was possible to grow embryos in cultures. With this recent discovery, Evans was able to extract embryonic stem cells from a mouse embryo, modify the DNA, and then reinsert it into the female.
Once the mice were born, all of their cells contained the modified DNA, and they were able to grow up and reproduce naturally.
Sir Martin Evans won the Nobel Prize for his discovery of the ability to modify the DNA of an organism in the early stages of embryonic development and have this modified DNA be present throughout the entire organism and still have the organism be able to thrive and reproduce naturally in its environment. The Nobel Prize did not go undeserved for Sir Martin Evans because even today we are still using the knowledge to better our advancements in the fields of medicine and biology.
One of the key benefits of this discovery is the knockout gene theory. This theory explains how you can change the DNA to knock out a gene. So you take the embryonic stem cell, you turn off a gene in the stem cell, and then the organism grows up and you see which trait is not present in the organism, and that tells you what the gene controlled. Right now, Sir Martin Evans is working with this knockout gene theory to discover which genes code for breast cancer. This is of particular interest to him because his wife, Judith, was diagnosed with breast cancer in the late 1990s.
As of right now, Sir Martin Evans knows the DNA sequences that code for breast cancer, and he is working on finding the genes that code for breast cancer. If he is able to find them, then he has the potential to eradicate breast cancer for future generations, and the possibility of helping his wife, Judith.
In 2007, Sir Martin J. Evans won the Nobel Peace Prize along with Mario R. Capecchi and Oliver Smithies, quote, for their discoveries of principles for introducing specific gene modifications in mice by the use of embryonic stem cells. Although this practice seems common today, it was a huge step in understanding more about genes, genomes, and their functions. Much thanks to Sir Martin J. Evans for making our scientific world that much stronger. And thank you for listening to our presentation.